So welcome to Click for IB Biology. We're in the transport section of the syllabus, and now we're dealing with capillaries. Now the capillaries follow the arteries in the flow of blood from the heart, so we're here. And what we're going to be looking at is how the capillary structure lends itself, is specialized for the exchange of substances with tissues. Now, should you wish, in the description there's a link to a PDF of this presentation, which allows you to annotate at your own pace. There's a quick quiz, and there are further channel links in there. So the blood has left the heart. It's under high pressure in the arteries, and we've already examined their structure. So when the blood arrives here at the capillaries, it's still under high pressure, despite the distance from the heart. The capillaries are a highly dense branching network, and this allows the blood to flow down many pathways, and so we will expect to see a significant pressure loss as it flows through the capillary. Now, the important consideration here is that nothing goes into or out of the blood in arteries, and neither does that occur in veins either. So this doesn't happen. Exchange only takes place in capillaries, and this is a significant learning point for many aspects of the course. So let's take a look at these capillaries. The typical cross-sectional diameter of a capillary is about 8 to 10 microns, and it's worth remembering that a red blood cell diameter can be around about 6 to 7 microns. So these are tiny, tiny structures. There are multiple cells. I'll just put some nuclei in here to illustrate this idea. And they're rolled up into this tubular structure. Around the outside of this tubular structure lies what is called the basement membrane. And this creates a protein-based collagen structure onto which the endothelial cells, we have it labeled here, the endothelial cells of the capillaries can attach themselves. Not all capillaries are the same, and this depends upon the junctions between one capillary endothelial cell and another. Some of them have end-to-end junctions like this. We call this continuous, and that would be typical of muscle cells and lung tissue. Others have gaps in them, which we call fenestration, and that would be typical of the kidney and the renal glomerulus, which is a topic we will return to later in the course. Others overlap in this discontinuous manner, and you can see that this is illustrated by the image of the capillary here, where we have this discontinuous overlap of the walls. As we move from the tight junctions of the continuous to the discontinuous, the capillary becomes increasingly, as the word here is, leaky. Its contents actually leak out into the tissues. This is a very important diagram for understanding diffusion and the exchange of substances between the blood and tissues. Here we have the capillary, and the blood flow is in this direction here. The pressure here is high, is coming from the arterial supply, and the pressure here would be low, which will be the venous end of the supply. Under the high pressure and the leakiness of the walls of the capillaries, fluid is forced out into the tissues, and we call this tissue fluid. Note that what remains inside the capillary are the cells and large plasma proteins. So the tissue fluid will contain all the dissolved substances such as glucose, amino acids and so forth, which are being transported by the plasma of the blood. So tissue fluid is derived from the plasma. So if we put that there, if we have plasma here, but to make tissue fluid, we don't have the cells and we don't have the plasma proteins. So what's left is largely water-based with dissolved substances. And so diffusion effectively takes place between tissue fluid and the cell in either direction, rather than directly with the capillary. Now, as the tissue fluid loses its dissolved substances to the cells, it becomes dilute. And this dilute tissue fluid is then attracted back into the capillary bed by osmosis. So it's forced out by pressure, but returns by osmosis. And in this way, we have a continuous circuit of the fluid from the into and out of the capillaries. However, the return of the tissue fluid isn't entirely 100%, and some is left within the tissue, and this drains away into another tubular system known as the lymphatic system. Now, the lymphatic system is composed of lymph vessels, as shown here, into which the tissue fluid, extra tissue fluid, will drain. And these converge on regions known as lymph nodes, and in turn, these empty back into the blood vessels. So ultimately, all of the tissue fluid circulates and is returned to the blood. This diagram here is critical not only for understanding exchange, but also aspects of immunology, which we will study in another section of the syllabus.